welcome to our 7th podcast in the information domain. In this one, we're going to spend our time with the process of meiosis. So here are our big questions. Ultimately, how is sex possible? Because we know with meiosis, it's the creation of gametes in order for two cells to fuse for the next generation. Why does it exist, and where does the variation in a population come from? So let's do it. So what it is, it's highly varied depending on the organism. Sex varies whether you're a small critter or a big critter. It varies, but at fundamental levels, the goal is the same. Genetic contribution from two parents to fuse together to create the next offspring. So that's why it's varied in terms of mechanics and the lifestyles. The overall idea, like I said, is the same thing. Okay, so whether you're talking about an animal, we know that we get gametes, fuse, go through meiosis to create the new critter, the fusion of two haploid cells. Plants and some algae have a little bit of some differences, so spend a time kind of looking at that. You get the creation of the zygote. You get meiosis, which creates ultimately spores that form into the gametes, and the fusion, and fertilization creates the next generation. And most fungi and protists really have a little bit of meiosis as far as how they form the fertilization of two gametes. You get your zygote. That zygote then creates the haploid cells. Okay, so really, with meiosis, we're creating haploid cells to be used to fuse together with the gamete or the haploid cell of another parent in order to create the next generation. So that variety, the life cycles among organisms really varies in the ways in which sex happens in order to create that next generation. However, a lot of organisms don't generally use sexual reproduction in order to produce. So they create clones of themselves, genetic contribution from one parent. You get an identical organism as the end result. So there's a couple of ways to create clones. Example would be like a budding hydra. So a little like sprout forms off the side, which we call a bud. That's the next offspring from that single parent. Sprouting redwood, this is from parts of the roots that kind of come around, and then a new plant, new tree sprouts from that. No genetic variation there. And then you have parthenogenic rotifer, and with parthenogenesis you get actually fusion of gametes, but it's still only one contribution from one parent. So yeah, a couple of ways that we make clones, those genetic identical folks. But in its sort of fundamental process, all sexual reproduction involves the same things. Take two haploid cells, create a diploid cell. Okay, we know this. And so just to remind ourselves a little bit on the terminology, haploid cell, that's your end count, that's your 1/2 of every chromosome pair, every homologous pair. That formation of the diploid means that we have 2 of every type of chromosome. And so most of our body cells are diploid. It's only the reproductive cells that are going to be haploid. So because we have different species with different number of chromosomes, you have different diploid and haploid counts. So example, humans, okay, we're 46. So for example, humans have 46 chromosomes, which means our gametes have 23. Fruit flies have 8. Their gametes have 4. Dogs, they have 78 chromosomes, which means that their haploid count is 39. So it's going to vary again, but notice those diploid counts are generally even numbers, right? So in order to do this and have a sexual reproduction and have that contribution from two parents, we need to make those haploid cells. And that haploid cell formation comes directly from a diploid cell from the organism. If we didn't go through that reduction process first, there'd be a problem in that there wouldn't be a consistent chromosome count in the next generation as far as what the right number is. We'd have an overabundance and craziness of chromosomes, and that would cause some pretty intensive problems. So meiosis, as I just mentioned, it's a reproductive for eukaryotic cell division, and there are stages of it that are similar to mitosis, but we're going to kind of focus in on the parts that are different. So because we have 2 copies of every chromosome, all of those copies are replicated, and we have our sister chromatids. So in one replicated chromosome, you have one replicated chromosome made up of 2 chromatids. Each of our chromosomes has a partner, a pair, because dad gives one, mom gives one. And we can see there that you've got, kind of the showcase of that karyotype, right? So here is, again, just hammering in that idea of what homologous pairs are, what a chromosome is versus a chromatid. Okay, so here is that duplicated chromosome linked together at its centromere. You've got one chromatid and its sister. So when you look at a karyotype, that is what you see, is an actually already replicated chromosome, because we're catching these either during prophase or metaphase. So what happens now? And just hoping, how about now? Are you getting this? Is this making sense? So if I have a cell that's 2n equals 6, right? So I have 3 pairs of chromosomes. In this cell, each chromosome has already replicated. I still have 6, right? I have 3 red, 3 blue, 6 chromosomes, 3 pairs, and we're already past S phase here. Okay, so this would be somewhere in prophase 1 of meiosis. So ultimately, the fate of all the chromosomes in meiosis is to break the pairs up, split them up, get them separated. So the stages here, you guys may want to just kind of pause, look at these, go back to the original presentation so that you guys can kind of play around with it. But just like in mitosis, okay, there is an interphase. You get an S phase where the chromosomes are going to replicate, but then once you enter meiosis, it's a 2-division process. You're going to get 2 actual splittings of the cell. So you get the first split, and then you get a second split. In the first one, okay, we'll spend a little time here, okay, because it's really during metaphase 1, well, prophase 1 and metaphase 1 that really vary between that and mitosis. In prophase 1, the homologous pairs actually pair up and they form a tetrad. During this time, they get so twisted and tangled with one another that you create a chiasmata, and that's going to be where crossing over is going to happen, where one chromatid and a section, it tangles with its non-sister chromatid, and they actually will flip sections. Okay, so in this case, what you're seeing is the red and the blue actually transferring segments from one another. This leads to further and greater genetic variation than just separating the pairs. So we get lots of variation that way. So here's that prophase 1 piece. That chiasmata that forms, you've got the centromere where they're connected, that crisscross of the chromosomes, the homologous chromosomes, is going to cause crossing over, and they condense, and you literally get this section that exchanges between the two. Okay, and that's how it works. So you can see here in this picture, you've got an exchange of information between the red and the blue chromatids, and the result is that you get very unique gametes, even more unique than what the parent had for recombination of genetic information. So back to here. So let's focus now on metaphase. During metaphase, those homologous pairs that formed the tetrads during prophase are going to line up directly down the center of the cell. That's still metaphase when chromosomes line up down the middle. This is very different than mitosis, because during mitosis, you don't ever see them pair up. It's just each replicated chromosome lining up down the center. So when each pair lines up, now we're ready to divide the pairs so that we no longer are going to have a diploid cell after the first division. Sister chromatids remain attached, though. Those replicated chromosomes remain attached, but we're going to pull the partners apart, and this is huge. Okay. Major difference. So pay attention to what this major difference is between the metaphase of mitosis and metaphase 1 of meiosis in particular. So take a second so that you can see those very distinctive differences that do occur through both of them. All right, let's take it continuing. Once we actually get that first split, and we've now divided our pairs apart from one another, there is no further DNA replication that happens. Okay, so we immediately enter into prophase 2. Now in prophase 2, we're going to get that, again, condensing of our chromosomes that are still as their chromatids. But in this one, instead, we're focusing on separating the sister chromatids from one another. So in this round, each chromosome lines up and each sister chromatid pulls apart, and you get at the very end, from starting with one diploid cell, I'm going to have 4 haploids. Okay, bear this in mind. These cells right here, the end cells from meiosis 1, they are haploid. Okay, please make sure you can understand and explain why that is. Why are these cells haploid versus the diploid parent cell? because once these split, again, my 4 that I get at the end of meiosis II are also haploid daughter cells. Okay, so at a glance, here's the deal. On this side, I've got a 2n equals 2. I've got a single pair of chromosomes. Okay, they replicate during S phase, form a tetrad. Okay, during prophase 1, meiosis 1, they're going to line up down the center of the cell. Okay, as anaphase 1 happens, that chromosome pair is going to get pulled apart, and you get the division where now each new cell contains only one of each homolog. So they're haploid. Now we head into metaphase, or excuse me, to meiosis 2. You get prophase 2, metaphase 2. Those chromosomes line up down the center, and we split the chromatids. And then at the end, the separation of the sister chromatids leaves me with 4 haploid cells. There we go. So major importance, or the 2 major importances of meiosis: produce haploid cells, create cells with new unique genetic information so that the combinations with the other parents' contributions create a combination that makes you unique. It's like taking the, we're just reshuffling the deck so that we get a new combination in the next generation and that they're not identical. Okay, so take a second here. You can spend some time. This kind of outlines, in terms of a summary, what are the similarities and differences between both mitosis and meiosis. So now, yes, now if only there were some strangely, awesomely cheesy and bizarre cartoon that would help make this all so clear.
One more time. Dana, hurry! We have to call up soon.
Okay, I'm duplicating as fast as I can.
Ah! There's the music. Quick, call up.
Call it time. Went to 2 miles, just right. And the phase 1 is almost done. Months is down, phase 1. The patient partners do their bow. Do see the cross over now. How many left with your left hand? Promenade around the land. Crossing on your legend, dear. Nuclear membrane breaks the near. Attention, Bobby, central Father's cold answer's done. Hold on down, it's metaphase 1. Couples moved in central plate. 10 phase 1, so we separate. Wait goodbye to reach your goal. Parkland moved to the other pole. Phase 1 is not your side. All alone, that's what you hate. Central plate from every two, slide before it's deja And in the phase 1 you duplicate, so that now you can separate. Father study, this is it, down the grow that tips to straight. This is just your normal state with some lucky appropriates. Rest a while and unwind you as nuclear membrane surrounds you. Now meiosis is complete for nuclei from one's a tree. Cytoplasm will be live, organics form with you inside. You will find many more the same kind. If you're wondering what's the use, also you can reproduce.
So there you have it. Cracks me up every time. All right, so why? Why do we bother with all of this in the first place? So really nobody knows exactly kind of where this all came from, but there is a sort of handicap that goes along with it. Now with asexual reproduction, you've got the female, you know, as we call them, that parent cell, we get daughter cells, and that constantly, generation after generation. Now in sexual reproduction, it's ultimately sort of the males aren't really part of producing the actual offspring itself. And so even with that though, it has evolved over time with many different lineages and so on. So it's kind of crazy. But the main big take-home message of all of this is that the increase to variation is exponential. It allows us to ensure that when we have kids, that our kids are not identical to each other, they're not identical to us, to help to promote the population generation after generation after generation. So huge, huge amounts, okay? And so in humans, right, so we have independent assortment, which means that each pair does not determine, you know, the separation of one pair doesn't determine how the pair separates in the other one. So the idea is that it's completely 50-50 which one of the chromosome pair heads in which direction. Okay, so you don't know whether or not, I mean, you're the gamete that made you from mom, you know, you could have had in that gamete, every single one of those 23 could have been the one, because of chance, the one that her mother gave her, or it could be a mixture of the ones that mom and dad gave her, and you got that mixture, and then the same thing with dad. So there are two possibilities for each of the 23 tetrads that form. Okay, so here's the deal. 2 to the 23rd possible gamete combinations, right? That's a whole lot. That's 8 million, right? But here's the catch. So random nature fertilization, getting one gamete to fertilize with the other. Okay, so if a male gamete, you've got that many choices. Female gamete, same choices, right? So that's really, per offspring, there is 70.3 trillion possible combinations per offspring. That is insane. Okay, so crossing over during prophase 1 enhances this. By the way, yeah, that 70 trillion, that is if crossing over doesn't even happen. So we can't predict crossing over. It's random. And so add to that 70 trillion what the possibilities for crossing over is that we can't even really calculate, and it's like, it's nuts. There's no way. As far as, you know, having a kid and then 3 years later having its identical twin, because of the way that the genes line up. So functionally, there are infinite possibilities of genetically unique offspring that we can produce from the same two parents, which is like nuts. Yet, let's have just a touch more on chromosomes before we finish up. Okay, so chromosomes we can look at because we can see them. This is the only time during meiosis and mitosis that we can see chromosomes, and we can put together and sort our pairs and get some understanding about what's going on in that organism based on how the pairs look. So pre-sorted, and then you sort them out and you find, and we create a karyotype. And based on the karyotypes, we can see whether or not there's any chromosomal abnormalities, which we'll look at in the next podcast, or and we can look at the actual sex of the offspring. So for all of us, for humans, we have autosomes. Those are pairs 1 through 22. They're the same homologous pairs, whether you're male or female, doesn't matter. 23rd pair, that's the sex determining pair. That's going to determine whether or not you're male or female. So if you're XX, homologous pair, you're a girl. XY, non-homologous pair, you're a boy. How she rolls, okay? So here are two normal karyotypes for a male and a female, and you'll note that last pair in the male versus the female. And it doesn't look like a Y. So there it is, meiosis. So in the next one, we're going to explore a little bit on what happens when the chromosomal counts go off, okay, and are not that 46 or not what they're supposed to be. So these are the things you want to make sure you can do. Have a great night, and we'll see you guys later.